video we're going to talk about regulation through variation in DNA, and the first one to it we have the access to DNA. In access to DNA we look at one of the two things. We do the acetylation of histone, or we can do methylation of DNA. So we can do either. Now what will happen, that there will be a loss of attractive charge and histone will leave DNA, and this is how we will have heterochromatin to form into euchromatin, right? This is done via HAT enzymes, and this is done in CpG islands where there is increased CG, cytosine and guanines, CG rich regions, and these are done by S-adenosyl methionine enzymes that acts as a donor, and we use different methylases to add that to the DNA. So SAM is the donor of the methyl group or the adenyl group or acetyl group, mostly the methyl group. All right, the next is amount of DNA. Now this is not seen in humans, it is mostly seen in non-mammalian animals. And actually what happens that when you increase gene amount or gene replication, the gene expression increases, right? But in case of humans, disease occurs. In case of humans, there is disease. Okay. The next is the arrangement of DNA. Now, arrangement of DNA, we have a good example of immunoglobulins. Let me write it in different. We have, okay, so then we have the arrangement of DNA. Now, in this example, we have the immunoglobulins. So what happens in germline cells, not in somatic cells, in germline cells, which means the sperms and the egg, okay, the sperms and the eggs, we have essentially what is called as three different regions. So we have V regions, V1, V2, V3. We have D regions, D1, D2, D3, and so on. And we also have J regions, J1, J2, and J3. So during crossing over or genetic recombination, these gene encodes in these genes, they will alternatively express and forms different types of immunoglobulins. Okay. The next one and the last one is the mobile DNA elements that are called as transposons. Now these transposons, they are mobile and they can regulate gene expression throughout the DNA. They can either move from one chromosome to another, so like these are two chromosomes and this is a transposon. It can move anywhere in this chromosome, or it can stand up from here and go sit there, right? But some transposons, what will they do is they will replicate themselves and they will sit there and then they will send the other one somewhere else. These are called as the These use reverse transcriptase enzymes, reverse transcriptase enzymes, and these are called as retrotransposons. Retrotransposons. This only happens in the eukaryotes, not in prokaryotes. And it also uses an enzyme called as transposase. Transposase. There is also an N. Transposase enzyme, and this is this is reverse transcripting activity of the retrotransposons. Now, in these are they mostly cause disease because they cause mutations. In prokaryotes, what happens is that they can cause antibiotic resistance because these transposons can learn to adapt, and they will learn the different active sites and what is the problem, where is this drug coming from, and they will edit the DNA or increase expression or alter the expression of the DNA. That's how transposons work. One thing in amount of DNA I will like to add is that if someone is taking methotrexate is an antibiotic or chemotherapy. It is a chemotherapeutic drug. It inhibits DHFR, that is dihydrofolate reductase. This is inhibited by methotrexate. But dihydrofolate reductase, it forms TTP, that is thymine, and thymine is essential for DNA synthesis. So what will DNA do? Because we need to make more DNA and we don't have thymidine, and we are giving methotrexate, and DHFR cannot, it's being inhibited. So there will be gene amplification for DHFR genes, and then we will increase TTP. But this will also cause resistance to methotrexate. So methotrexate is then obviously given quite carefully with dosages managed, and of course with folic acid and all that. So yeah, that is it for today. Thank you so much.